I'm sorry. Instead of knocking off the two.
Test, test, test. Test, test, test.
That's what it should show. That's what it should. I changed the webcam to the top one. So it's just.
Unmute. And so welcome to our symposium, our fall senior symposium. It's exciting to see some faces like Lily that I haven't seen for a while. And so thank you for coming to support our seniors. And I just am going to open us up with a word of prayer, and then I'll give a couple of very brief instructions. Heavenly Father, we thank you for who you are. We thank you for this great creation that you have made, and for the opportunity that we have to study it a little bit. And I pray for these students, and I'm just so thankful for the opportunity to work with them and get to know them a little bit. I pray that you would calm any nerves that might be present and help them to do well as they present. And I pray too for their future endeavors that you would just go with them as they leave Bethel. And I thank you and praise you and just commit this time to you in Jesus' name. Amen. All right. So we are going to try to stay on schedule. So we're already 2 minutes late. So this is the way I normally run things, a little bit late. So we're going to try as much as possible to stay on schedule. And our first presenter, okay, before I say that, I will try to notify you if it's time for you to wrap things up. Does that make sense? And maybe I'll give you like a 5-minute warning, and then maybe I'll wrap it up kind of warning. Okay. And Our first presenter is Molly Longton. She's going to talk to us about the impact of goat grazing of Rhamnus cathartica. I think what I'm going to do is I'm going to kind of minimize that. That up there. Hopefully that'll maybe down here. And then take it away, Molly.
All right. Okay, I'm really excited to start us off in the ecology room. I'm going to be talking about the importance of goat grazing of Rhamnus cathartica European buckthorn on percent cover. Oh, it's not working. Okay, it was working earlier.
Oh.
That's okay.
Oh, maybe you have to
Click on the
click
screen.
on the.
Oh, there we go.
All
All right. Let's get started. Okay, so what is European buckthorn? Buckthorn was introduced to North American ecosystems in the early 1700s, eventually being transported to Minnesota in the 1850s to be used ornamentally, so in parks and gardens, just kind of, people liked the way it looked. It's now found in nearly every part of our state because of many of its vegetative characteristics. So it likes to be kind of a monopoly in the ecosystem that it resides, kind of dominating nutrients, light, moisture content. It's easily spread via avian species, and it has a really extensive root system with virtually no predation or disease susceptibility. It also serves as a host for other invasives like crown rust fungus and the soybean aphid, which is a really big problem for our crops. So how has it impacted our ecosystem? Like I said before, buckthorn, when it's introduced, it completely dominates the ecosystem. And just the overall ecosystem function. When buckthorn is present, when it's present, there's a lack of older trees because our native species are pushed out, which then leads to a decline in wildlife habitat, which kind of, furthers the domino effect, providing a high stress environment for animals and plants, leading to a decline in reproduction, a decline in population, and ultimately what we're seeing, a loss of biodiversity. So how do we control it? This is the big question. There are two main groups of buckthorn control, chemical treatments and non-chemical treatments. Both have shown to be effective to a certain degree. For chemical treatments, you can use herbicides containing trichopter and then herbicides containing glyphosate. For our non-chemical treatments, we have mechanical control, which is going in, cutting up the stands, picking up the newly emergent seedlings, and kind of just manually removing the buckthorn. Another most recently researched way of treating buckthorn non-chemically is goat grazing, which is what I'm going to be talking about today. So this is my research question. Is goat grazing an effective method of reducing overall percent coverage of European buckthorn in the Listener Woods on the Bethel University campus? You can see my hypothesis. Initially, I thought that there would be a significant decrease in the overall percent coverage of European buckthorn in plots grazed by goats compared to ungrazed plots. So this is how I did that. These are the plots that I took data from. You can see they are highlighted. I used plots A and B, which are my control plots. They're not grazed by goats. And then I have plots C and D, which were grazed by goats. This map was made by Jessica Lilly. She started the initial goat grazing study, and she went out and divided these plots, kind of just made this whole study. So this is how she did her data collection initially back in 2018. She divided the plots into equal acres, so they are all 0.66 of an acre to provide uniformity in collection. When deciding where on campus she would do this study, she factored in three criteria: a uniform presence of buckthorn, a lack of disc golf holes, those are really popular on campus, and a lack of sprinklers, because both disc golfs and sprinklers would cause a disruption in goat grazing. For her data collection, she did 3 rounds of data collection. She initially went out, took photos, which we can see on the other side of the presentation slide, which is a 1 by 1 meter square used to outline the sample plot visually. But she did that for May, which is before the goats came. She did that in June when the goats had grazed the first time, and she did that in September when the goats had grazed a second time. So for each plot, they had the frame, which was used in every single plot within, or every single data collection point. And then it also had all of the collection information, which included a random number that was assigned. So this one is A3. It had the date that it was collected, the time, and just any other information that was relevant to that. So for my analysis, I chose to analyze the photos in using image analysis software, ImageJ. And this is where I chose to use the steps to kind of isolate the leaves from the background of the photo so that I could just see like if there is a difference in percent cover. This was done for all of the ground images for each plot, A through D, pre-grazed, post-grazed for the first time, and post-grazed for the second time. So this is how I did it, kind of a visual representation. The yellow line is kind of hard to see on the initial image, which is in color, but it was, I outlined the 1 by 1 square using a selection tool, factoring in specific criteria. So that included plants that originated from the outside of the plot, I chose to exclude, and then plants that originated from the inside of the plot, I chose to include, which you can kind of see over here. I included some of those leaves so that it was uniform in the plot. I then altered the image, which is a color image, to an 8-bit image to analyze the grayscale. So this is when I took initial measurements, the area of the plot, the max grayscales and gray values that I could get, and then the minimum gray values, so that I can compare that with my data after I changed the threshold. So this is where most of my data came from, was when I changed the threshold. So this is so that you can isolate the leaves from the forest floor. So you can see all of the gray in this image are just leaves, and then all of the red is the background. So it was recommended from a few different sources to select the dark background and then not to reset the range. Otherwise your data is kind of skewed. And then after this, just to get more measurements, you have to make sure that you reselect the original selection. So otherwise your data is whack. So again, I ensured that the original selection was highlighted, that all the values, you're getting like a whole image value, and it just messes with the whole thing. But you can see that it's back in just the binary image. So you can see that all the leaves are black values and all of the background is the white values. And from this, I can analyze a histogram, which shows just the distribution of the black and white values in the active selection. So from this data, I calculated canopy cover. This formula was from the United States Department of Agriculture Forest Division, where the mean, so the M, the bolded M, is the mean black values within the selected area of the binary image with the altered threshold. So this is what I got in the later steps, the step with the red. And then the capital M was the max number of potential pixels, which is within the selection area of the binary image without an altered threshold. So that was the initial measurements that I got. From this, I could do some statistical analysis, average percent cover for experimental plots and control plots, descriptive statistics, and then I also did a t-test. I did a two-tailed t-test for all 4 plots. So my first t-test was comparing May data with June data. So pre-grazed with post-grazed for the first time. And then my second t-test was pre-grazed with post-grazed for a second time. So I was comparing pre for both sets of data. This was done for all of the 4 plots. Here are my results. So as we can see, this is a comparison of pre-grazed to post-grazed 1 and post-grazed 2 for both the control and experimental plots. The control is highlighted in green, which is a little difficult to see on the projector, but it's on the left and then the experimental was on the right. We can see that there was a decrease from pre-grazed to post-grazed 1. But we can see that it's a little bit different for post-grazed 2. It's kind of the same for both experimental and control lots. Here are my p-values. This is what I got with my t-tests. Unfortunately, for all of the plots, they were not statistically significant. All of them were above 0.05. I did have a few that were like on the brink, but for plot B, that was an ungrazed plot, so it just wasn't statistically significant. This is kind of highlighting how we saw an overall decrease. So this is just the decrease from pre-grazed to post-grazed for control and experimental plots. So we can see that there is a higher % decrease from pre-grazed to post-grazed 1 than from pre-grazed to post-grazed 2 in our experimental plots. So we do see a little bit of difference from grazing versus ungrazed. So let's talk about this. Canopy cover is ultimately not different before and after go grazing in 2018. Although there was a % decrease in the grazed plots, ultimately it was not statistically significant. However, we can kind of see that in the photos. So this is outlining C14, which is a plot within the grazed section. You can see in the top that is the pre-grazed. This is before goats came. We can see that there's a lot of vegetation within the 1x1 frame. The second photo is post-grazed for the first time. This is in early June. And we can see that there is a decrease from the first time to the second, from the before and the after. But we can also see that for post-grazed 2, which is the second round of goat grazing, we did see a little bit of an increase in vegetation. So that would make sense for our decrease to be a little bit different because we do see an increase in our graze plots. So why is there not a decrease in % cover? Ultimately, goats are really picky when it comes to their food. There's definitely a stereotype that goats will eat anything, but ultimately that's not true. They only like to eat stems that are 30 to 150 centimeters in height. And all of my ground images included stems that were under 30 centimeters. So ultimately I didn't analyze the stems and the plants that they wanted to eat. This is just something they wanted. So let's also talk about some challenges that I encountered with my data. So there was a little bit of an inconsistency in photographs. The time of the day that photos were taken, light availability, angle that the photos were taken at, and kind of just the organic material obstruction. So some plots had a boot in them from Jess Louise, her partner. Some had a big, big logs and stuff that kind of skewed the data values. Also, there was a bit of an inconsistency in image J. So there was a high concentration of dead leaves, which the image analysis software did have a little bit of a harder time distinguishing between dead leaves and living leaves, just because when I'm analyzing it from a grayscale value, they do look a little bit the same. There was also some other challenges, like missing photographs. Some plots had pre and post and not post for the second time, and some had pre and then post too. So there was some missing data. And there's also an inconsistency in plot vegetation. Some plots were very deep woods and some were in the grass. So that has a lot of inconsistency when it comes to analyzing visually. And also goats don't like to eat stems that are small, like I talked about before. Ultimately, I think that additional research is needed for a decrease in lower canopy cover. If I was to repeat this study, I would go with some more consistent photographic methods. Ultimately, when Jess went out and took these photos, they weren't intended for image analysis. They were just intended to kind of highlight the data that she was collecting. I'd also ensure that there's no absent data, comparing plots with a little bit more similar vegetation, and then just doing some additional analysis regarding trees that are 30 to 150 centimeters in height. So maybe taking photos from a higher angle, seeing what that would bring us. I have to acknowledge my research supervisor, Dr. Weiss, who was on Zoom today, and Jessica Lilly, who did the initial data collection. Hannah Dansigal, who also was after Jess and did a lot of the methods, and kind of outlined what Jess did in her data study, and then everybody else that collected the goat research. And here are my references. These are but my-
We'll take time for one question.
Sorry.
It's not your fault.
All right. Any questions?
How do you figure out how to use ImageJ?
ImageJ is a really difficult software. I don't know if anyone has used that software. It took a lot of trial and error, just analyzing the different ways that I could like look at the image. Because it's initially ImageJ is really kind of used for like cellular image analysis, so looking at like gel electrophoresis and stuff like that. So it's different, but people have used ImageJ to analyze canopy cover, but it was kind of just trial and error. So just figuring out, what worked best. So yeah, awesome.
Thank you very much.
Thank you.
Good job, Molly.
All right.
I think I'm gonna close yours, Molly.
Oh yeah, totally. Right. She's gonna present again. Actually, I'm gonna present. That's the only thing.
Our next presenter is Anna Stockville, and Anna is also going to talk to us about the effect of goat grazing on Rhamnus cathartica. Perhaps a little
Overlap maybe.
overlap, and maybe you approached it differently, and we're going to find out.
Okay, thank you, Dr. Dykstra. So, hi, my name is Anna Stockdale, and I will be discussing my research on the effect of goat grazing on Rhamnus cathartica. Which I apologize ahead of time. There will be a little bit of overlap in Buckthorn history, but that's just the nature of it. So, Buckthorn was introduced to North America, more specifically Minnesota, in about the 1800s, and it took approximately 120 years to actually identify it as an invasive species. Now, as you can see on the bottom right is a map of just showing a little bit of buckthorn distribution as of 2016. Sadly, I could not find an updated map. However, it kind of just puts the point across that I needed to. And as you can probably tell, buckthorn mainly grows in deciduous forests as well as woodlands. So what is the effect of buckthorn on ecosystems? Well, since it is an invasive species, it has an overall negative effect, and that can be seen because of its wide seed dispersal. Buckthorn produces berries that birds eat, and then they fly like further away, excrete the seeds, and they can germinate then a lot further away from the parent plant, which is a different version of seed dispersal than a lot of other plants. So that is used for their advantage. They also can limit growth of native species due to the fact that they leave at a very early time in the spring, and that can prevent a lot of native species from obtaining sunlight that they need in order to grow. And then this graph on the right, I actually made in my ecology class, and it shows the relative abundance of adult and young woody species on Bethel campus. I just thought it was relevant because it kind of shows, like on our campus, how much buckthorn has really taken over. All right, so there are a few different ways of controlling buckthorn, one of which is actually cutting stems to a certain length in order to try to kind of stunt its growth. Another is herbicide treatment, controlled burning, and then last is biological control, like goats, which is what we'll be talking about. All right, so my overall research question was, what are the long-term impacts of goat grazing on populations of Rhamnus cathartica? Excuse me. My hypothesis is that 2 years after goat grazing, there will be, in a Midwestern woodlot, there will be a significant difference in buckthorn growth between grazed and ungrazed plots. Ultimately, I wanted to see less buckthorn in the grazed than the ungrazed plots. So my methods were established, like Molly talked about, by Jess Lilly in 2018, and she created this beautiful diagram on the bottom that shows 2 control plots, which were ungrazed, and then 3 experimental plots which were grazed. That's where I collected my data. All right, so there were about 60 different areas within the plots that were marked by this bright pink stake that used a little GPS to like walk through all of the vegetation to find. Once you finally found it, you put down a 1 by 1 meter PVC pipe square onto the forest floor. And then you count the number of buckthorn stems that were above 30 centimeters, as well as ones that were below 30 centimeters, making sure to not count any that were under the square or outside the square. Data collection was pretty quickly between the dates of June 1st and 9th of 2021. And then I compared the data of this year to 2020, 2019, and 2018. Along the way, took a few photos such as this one to kind of represent, do a visual representation of growth. All right, here's 2 examples of my data collection once I had it organized in a spreadsheet. And you might notice that there are some dashes for plot numbers, and that represents some plot numbers that we could not find. Did a lot of searching, but I don't know, maybe the stake was broken off or lost some way. There were 5 of those found in the ungrazed and then 5 found in the grazed, which would impact the data analysis. However, it's kind of nice that it was 5 in both. Then I performed a t-test and a 2-factor ANOVA for data analysis. Here's just a little bit of observational data because there is so much variation between different areas within the plots. There are some squares that were full of buckthorn stems, like greater than 30 centimeters, less than 30 centimeters. And then there are some that had no stems at all. And I wanted to make note of that because that's actually pretty significant. All right, so here are my results. First off, looking at the total number of stems counted in 2021. Obviously, you can see that the number of stems that were less than 30 centimeters is greater, which makes sense because we went through the goats 2 years ago, so they can only grow so much in that amount of time. My p-value for less than 30 centimeters was 0.56, and then for greater than 30 centimeters is 0.55, which sadly shows that there's no statistical significance. So here's a little comparison of previous years. There's a lot to unpack here, but we're gonna do it. So looking at less than 30 centimeters, between 2018 and 2019, there was an overall increase in the ungrazed plots and then a decrease in the grazed plots. For greater than 30 centimeters, there was an increase in both. And then go grazing actually ended year 2019. However, we still saw a dramatic decrease in the number of buckthorn stems in 2020, which I'll cover reasoning a little bit later. Looking between 2020 and 2021, there was a decrease in grazed and an increase in ungrazed, just like in 2018, 2019. And then for stems greater than 30, it was an increase in both. So there's that. Here are the results of my ANOVA test, and it shows that there is only significance between the data collected in 2018 versus 2020, and then 2019 versus 2020. No other years were significant. All right, so some conclusions. There is no statistical significance between grazed and ungrazed plots 2 years after go grazing. But time is the significant predictor, and 2020 was an anomaly. So why was 2020 so different? Other than the fact that all of our lives were dramatically changed from COVID. I actually looked into it, and there was an early freeze in 2019, which would have negatively impacted the germination and then growth of all plants in 2020. We've also experienced droughts in both the year 2020 and 2021. Of course, you also have to take into account that there are other environmental factors such as precipitation and climate change, that those are a lot harder to take into account and calculate, and that's sadly was not part of my research. So what if 2020 was a normal year? Well, we would have expected to see trends from 2018 and 2019 to continue last year and this year. Are goats a viable solution? That's the big question, right? Statistically, no. However, it's a lot more complicated than that. And we still did see a decline in buckthorn population, which is the ultimate goal. Looking forward, I would continue with methods and consider doing 2 years of goat grazing and then 2 years off of grazing and kind of continue that for several years. I'd also take into account other environmental factors, and I would like to mention the fact that there are only 2 control plots and then 3 experimental plots, because we originally started with just 2 and 2. Yeah, we had to add the 3rd one because the goats were going through everything pretty quickly. And so I would consider making plots in a new area, new plots, and having a consistency of controlled and then experimental plots, same number of both. Okay, here's my works cited, and then I would like to thank Dr. Wise, who's on Zoom, for being so amazing and helping me through this whole thing, as well as my lab partner, or my research partner, Joey, who's sadly not presenting this semester. You got to come back next semester, but his research will also be on around like the same area, so check in for that. Any questions?
I'll start with the questions. I was wondering, with the plots that you couldn't find, the quadrats that you couldn't find, I wonder if you looked at whether they were sort of influential in like the increases or decreases that you saw versus what your data. Do you understand my question?
I think so. I didn't look into it too much, just because there were a lot of other areas that we went to, like the plot IDs or whatever. So I didn't take it into too much consideration, but that's a really good question. Thank you. Yeah.
What was your reasoning for like you wanted to look at 2 and 2 rotation potentially in the future? What's your reasoning behind that?
I noticed that the effect of buckthorn goat grazing has kind of gone away after 2 years. So I just feel like, and it'd be a good time span to do like grazing and then not grazing. Also, because we only grazed for 2 years, so if we keep it 2 and 2, that'd make things a lot simpler, in my opinion.
Other questions? Okay, thank you again.
Okay, thank you. Sorry.
Our third presenter of the night is Daniel Dreschel. Did I say that right? Dreschel. Apologize. And we're going to hear another talk about European buckthorn. Only this time it may not have as much to do with goats.
Hi everyone, as Dr. Bex just said, I'm Daniel, and I also will be speaking about buckthorn. So you've already heard this, but a little bit of background about buckthorn. It's a persistent and continuous invasive species that is found here in Minnesota and also much of the greater Midwest and northeastern parts of the United States. And it was originally brought here as an ornamental decoration from parts of Europe and Asia. However, it quickly grew out of control and now can be found in a variety of habitats which include fields, forests, which is where it is primarily found here on Bethel's campus, and even anthropogenic areas, which are areas that have been impacted by human pollution. And this spread by buckthorn is due to a variety of ecological advantages it has over a native competition. The first one is that it is able to survive in both drought and flood conditions, which, as we saw this summer with the recent drought, that can be very advantageous for plant species. More importantly, however, it is tolerant to deep shade, and this is largely due to the fact that buckthorn can leaf out earlier in the spring and maintain its leaves longer into the fall. And actually, buckthorn can maintain its leaves about 58 days longer than other local competitors. And during that time, buckthorn is able to take up 38% of its annual carbon intake. Buckthorn can also negatively impact the soil, in particular soil composition. Studies have shown that where there's a large amount of buckthorn, there can be up to an 80% increase of both nitrogen and carbon in the soil. And this is largely due to the fact that as buckthorn leaves decompose, nitrogen either goes into the soil or is released as a greenhouse gas. And this can lead to elevated pH in the soil. Studies have also shown that buckthorn can be allelopathic, which means that it can produce chemicals to suppress other plant life around it. As a result of the spread of buckthorn, many control methods have been implemented to try to curb buckthorn populations. These include biocontrols, chemical controls, which include herbicides, as well as mechanical controls, which is going out and cutting down buckthorn. As a result of that, and as a different way to control buckthorn populations, Bethel has brought goats here on to our campus, and studies have shown that it can be a competent way to at least initially suppress buckthorn populations. However, those same studies also show that goats tend not to eat young buckthorn, in particular those younger than 30, shorter than 30 centimeters in stem height. So the goals of my research were a little bit different. And the primary goal was to establish a control and experimental plot of land for further research into looking at goat grazing. And to do this, I hope to quantify the presence of buckthorn in the Seminary Hill Woods here on Bethel University's campus before any goats grazed upon the buckthorn. I also hope to gather data on the pH of the soil and identify any other invasive species that could come in if we successfully remove buckthorn. To do this, I first used a Trimble Geo 7X to go out in the woods and outline the Seminary Hill Woods in the area that I wanted to do my research in. I then used Microsoft Excel to select 40 random points within that area that I outlined, and then divided the overall outline into 2 smaller areas, each with 20 points each. These were then labeled plot A and B respectively. It should be noted that I controlled to have 3 points in both plot A and B to be near the water's edge. This is a map showing the overall outline of my research area, and as you can see, it's next to Bethel Seminary and right on the edge of Lake Valentine. And as you can see, pen A is the northernmost plot and pen B is the southernmost. Once I established the 2 plots, I then used the Trimble again to go out into the woods and find each of those random points that I generated. I then marked each point with a stake and labeled it with the plot name, A or B, and the number 1 through 20. I then used this 1 by 1 meter PVC pipe outline to outline my data collection area. I then counted the number of buckthorn in that area and divided it into 3 categories. Those less than 30 centimeters in height, those 30 centimeters in height to diameter breast height, which is roughly 160 centimeters, and those greater than diameter breast height. And if buckthorn plant was greater than diameter breast height, the circumference of that buckthorn was taken. Additionally, I also collected data on the pH of the soil in that quadrat, as well as the number of other woody vegetation, the number of forbs, the number of graminoids, the presence of non-vascular plants, as well as any other invasive species in the area. And I also noted the presence of any wood chips within that plot. I then used a 2-sample t-test in each of these 4 categories to see if there were any statistical differences between plot A and B. And the 4 categories are buckthorn 0 to 30 centimeters in height, 30 centimeters diameter breast height, and that greater than diameter breast height. And I also analyzed the mean pH by 1 meter quadrat by plot. And my results showed that while there were differences between plot A and B in the mean number of buckthorn in each 1 meter quadrat by plot, none of the differences were significant. And as you can see, the p-value in all 3 categories was greater than 0.05. So as a result, we can conclude that in buckthorn density, both plot A and B are equivalent. The same is also true of mean pH by 1 meter quadrat by plot with the p-value, although it is being lower, is still greater than 0.05. So again, there is no statistically significant difference. So what does this all mean? Firstly, as I said, there is no difference in any of the 3 categories between plot A and B in the 3 categories of buckthorn between plot A and B. And that is particularly good for my research because I hope to establish a control and experimental plot for further research. So the fact that they're statistically not different is good for that. There also seemed to be a negative trend between the number of buckthorn and the size of buckthorn. This could be accounted for a variety of reasons. The first is that buckthorn has a large seed bank so that it deposits seeds into the soil. And if we were to successfully remove buckthorn, the new young buckthorn would spring right up and take its place. It also speaks to the adaptability of buckthorn and that buckthorn will grow up wherever a space presents itself on campus. And it also makes sense with the general life cycle of buckthorn, which states that as buckthorn increases in size, it'll take more resources to support that. So it'll tend to be a lower density of buckthorn as they increase in size. These are some other environmental observations that are not quantifiable, but are rather just observational data that I noted during our research process. First, there's a large presence of goldenrod, solidago, along the edge of the woods. And although this is an edge species, if we successfully remove buckthorn, large open areas could open up that goldenrod might be able to spread into. There also seemed to be a positive trend between the waterfront and buckthorn density. So more buckthorn was present near to the in those quadrats near to the waterfront. However, I only have 3 quadrats in plot A and B that were on the waterfront, so I didn't have enough data to run any statistical analysis. The same is also true of wood chips. I was interested to see if wood chips would have an effect on pH, but wood chips were only present in a few of my quadrats. So again, not enough data was present. The time of year also could have affected my data collection, as I collected it in August and September. There was likely less vegetation on the ground overall. And there was a drought last summer, so overall there was less vegetation throughout Minnesota. More importantly, however, because of mechanical removal of buckthorn in plot B has larger open spaces with lots of young buckthorn just starting to spring up, whereas plot A has a more consistent spread of buckthorn throughout its area. And as a result of that, I proposed that for further research, plot B be established as an experimental plot and plot A be established as a control plot. And this is of particular interest to us because, as you saw in the other presentations, goats tend not to eat buckthorn less than 30 centimeters in height. So if plot B is used for further goat grazing, there will be more research and more data available probably in the future to determine if goats will eat that 0 to 30 centimeter buckthorn. So as I said, more research needs to go into the fate of buckthorn, especially young buckthorn if goats are present to eat that. But also more research should go into effect and correlation between buckthorn, wood chips, and pH. If there is any effect here on Bethel University's campus. And we also need to continue to monitor influx of any new invasive species that could take buckthorn's place if we successfully remove it. And last but not least, more research could be done to determine if goat grazing is more effective if it is paired and coupled with mechanical removal of buckthorn. I'd like to thank Dr. Sarah Wise, who's on Zoom, but I would not have been able to do it without her. And I also like to thank Dr. Amy Dykstra for her help with the Trimble, but also the entire biological and environmental science programs here at Bethel University. So my references. Thank you. Are there any questions?
They just have 2 plots that you established, right?
Yep, just
How many like samples would you say like you would recommend taking for data?
in the future.
each plot? Yeah.
I would say it probably depends on the project. But I would say at least 20 points would probably be a good amount for each plot, as they're both pretty large in size and they have a lot of differences in composition throughout the whole area. So I would say you would need a larger sample size just to take into account all those differences in composition.
And then, what did you say was the observed effect of waterfront on buckthorn?
So in my quadrats that were near the waterfront, there appeared to be more buckthorn.
What do you think that was?
I'm not sure. I didn't look into it too much, but I assumed there tend to be more vegetation around waterfront in general. So I assumed that could have an effect on it. But I didn't look into it too much, as I only had a couple points near the waterfront.
I mean, you noted that there were differences in terms of some of the larger buckthorn between A and B, right? There's been some facilities that's gone through and cut down some of those large seed-bearing trees. Was that reflected in any way in the quantification of each of those plots, or how would you kind of factor that into the decision to? Basically, what I got out of that was, I think it was A really was a more uniform distribution, and B was a much more patchy distribution of buckthorn because of the removals.
Sorry, what was your
Yeah,
question?
so how would you sort of account, or how did you account for those differences between A and B? Because that in itself is potentially a difference.
Sure.
In a control versus an experimental setup.
Sure. Yeah, there were differences, but still, there were areas with large amounts of dense buckthorn and plot B. And so in my t-tests, there was still no significant difference because well, there was like larger variance in plot B, but the mean still came out to be roughly equivalent to that of plot A, if that makes sense.
It does. So, but to use some of the terms from ecology that we were talking about, right? So you're talking about dispersion differences.
Sure.
Right? So the distribution of the plants within the two are very different.
Yeah,
Or
the
at least-
dispersion is different, but the abundance too is.
Thank you.
Take it again.
All right, thank you.
I should have closed this door like an hour ago, but I didn't. Okay.
Pointer should work as long as. So let me just go down. So this one's yours?
Yes.
Okay. So. It should work now because the screen that's on the front is your presentation. So it should work.
Our next presentation is Erica Swanson, and she's gonna mix it up a little bit. Talk to us about birds. So we're delighted. Careful with those cords.
Well, hello, everybody. Thank you, Dr. Dexter, for the introduction. And yeah, I'm going to talk about birds because for those of you who don't know, I love birds. I've been a bird watcher since I was like 3, 5 years old, somewhere in there. So a long time. I have been involved in various bird research since my sophomore year of high school. And one of the topics I'm most interested in is human impacts on bird populations. So for this summer, I wanted to focus on the rush hour traffic noise and how bird populations respond to that. So when we look at overall biodiversity, there have been lots of studies done that show that humans can have an impact on this biodiversity. For example, we see a decrease in biodiversity from this infrastructure, such as windmills can disrupt various populations. When it comes to birds in particular, we have seen edge effects, isolated populations, and increased mortality. So what each of these means, so edge effect is when you have a habitat and infrastructure is immediately adjacent to that. At the edge of the habitat, the infrastructure has a greater effect than when you get further into the habitat. Isolation of populations is sort of the same as that, where you have some type of infrastructure that cuts the habitat into multiple sections, and this can make it difficult for animals to cross that barrier. Also, we have seen an increase in mortality in avian populations. Again, this would be like with windmills where birds get hit by that, or I'm sure you've all seen a bird hit a window of your building. Also, birds can get hit by cars, which is very unfortunate. And when it comes to traffic noise, there have been studies showing that this can mask important sounds. So for example, a bird would not be able to hear a predator approaching as easily as it would without the noise. It can also disrupt communication between birds, so the birds cannot communicate as effectively with each other. So overall, we can see that understanding the effects of infrastructure can be important to understanding how we can better improve our conservation of wildlife and preserve nature for the future. So to look at traffic noise effects on birds, I looked at some previous studies and found that some showed a decrease in abundance, but some showed no negative effect. So because of this variability, I wanted to bring clarity to these results, but also investigate short-term responses. So all the studies that I looked at and all the studies I could find were on traffic noise in general. So whether that's just looking at an edge effect of traffic or looking at if you build a road in through a habitat and looking at the effect of noise specifically in that, there was not a short-term effect. So to do that, I wanted to look at rush hour specifically. So is there a difference in abundance when you look at bird populations during rush hour and then outside of rush hour? So to set up my experiment, I chose 4 parks, all of which are part of the Three Rivers Park District, which is northwest of the Twin Cities. These parks were Elm Creek, Fish Lake, Crowhassan, and Baker. I set up 3 transects at each of these, 50 meters from the road, 250 meters from the road, and 450 meters from the road. There were 8 points along each, which I marked with flagging tape, as you can see here. Oh, that went back. Okay, I will not be using the laser pointer. Anyway, back to this. We stopped at that point. So then I collected data at each park for a total of 8 weekdays and 4 weekend days for each park. And then I had to analyze the data. So as I said, I had 4 parks. Crowhassan is on the left, Baker and Elm Creek are shown in the middle, and Fish Lake is on the right. I chose each of these parks because there was a significant difference in traffic for rush hour times and non-rush hour times. So to do this, I would use my weekdays as the rush hour time, and then the weekends as my outside of rush hour time. As you can see, there are some habitat differences in each of these. For example, Crowhassan's that had some wooded areas and then also some prairie areas. Elm Creek was mainly sparse trees. Baker was sort of the same as Crowhassan, where there was a lot of wooded areas, but there's also some prairie. And then Fish Lake had a variety of habitats from wetlands to woodlands. So here are the maps of my transects. I have the roads marked. So Crowhassan was along County Road 19 in Hanover, and As you can see, the 50-meter transect is closest to the road, 250 meters is in the middle, and then 450 meters is the farthest. This is the same for other parks as well. Also, as you can see, I tried to follow the contour of the road as best as I could. There were some habitat challenges with this, such as wetlands. I did not want to just walk through wetlands all the time. I got waterproof boots, which helped, but only waterproof to some extent. They're also only waterproof from the outside. Once you get water in them, they're not waterproof. So anyway, as I walked along these train sects, I would alternate which side I started at. So 1 week I would start the 50-meter transect closest to the road and then move into the habitat and end at the 450-meter transect. The next time I visited the park, I would start the 450-meter transect and work my way back towards the road. This would control for time, since all of my visits were between 6 and 9 a.m. on weekends and weekdays. When I walked along the transects, I would collect data. So at each point, I would stop for 5 minutes and collect a decibel level to analyze the traffic noise. I would count individual birds and also how many species were present. When I was at the 50-meter transects, I would also take a traffic count of a sample of 1 minute at each point, so I could average the overall traffic and determine the traffic flow volume. So to look at my results, I compared my mean decibel levels and then also my mean avian abundance between each transect at each park. So the graph, or the table on the left, shows my comparisons of each transect for each park. This is looking at abundance. So as we moved from the 50 meters to the 450 meters, between each transect, there was a significant difference in abundance. There was also a significant difference in the traffic noise. The only one that didn't have a significant difference in abundance, I'd like to point out, was Fish Lake when comparing the transect 250 meters from the road and 450 meters from the road. Again, this is looking at all my data. So this is weekdays and weekends combined, just looking at differences between the transects. On the right in the graph, we have abundance plotted in blue and mean decibel level plotted in gray. So again, as we start closer to the road, that abundance is lower. And then as we move away from the road, the abundance increased. And then for decibel level, as we moved towards the road, or as we started near the road, it was higher, and moved away from the road, it got lower. The second one that we looked at was a traffic noise effect. So to do this, I looked at my weekday data versus my weekend data. I compared data points from each of the transects at each of the parks. What's important to note is on this graph, the abundance is what we're comparing, that's blue and gray, and then we're also comparing the yellow and orange. So there was a significant difference in the mean decibel levels from weekend to weekday, and that's shown again in yellow and orange. And then there was also a significant difference in gray and blue, the abundances, but this only was significant at Elm Creek at the 250 transect and the 450 meter transect. So as we went away from the road, that increased, but the weekend abundance was higher than the weekday abundance, and the weekday mean decibel was higher than the weekend mean decibel. Basically, this means that it was louder during the week on the rush hour days, and it was quieter when there wasn't as much traffic. And also we saw more birds on the weekend at Elm Creek for the 250 meters and 450 meters. We saw this trend in most of the parks, but it was not significant in all the parks. So to summarize, we saw an increase in decibel level as we moved towards the road and a decrease in abundance as we moved towards the road. So this suggests an edge effect because there were fewer birds close to the road where the traffic noise was highest. As we moved into that habitat, that effect decreased. Also, as I mentioned, there was a difference at the 250 meter, 450 meter transects at Elm Creek. So this suggests that there was a traffic noise effect, specifically a rush hour traffic noise effect at these transects. So why only those two transects? Well, one thing to notice was when I was at Elm Creek, the 50 meter transect, I didn't see very many birds there at all, whether that was on weekdays or weekends, so that difference was not very great either. Also, Elm Creek Park had the highest traffic flow compared to the other parks which did not. So during the week, for the rush hour data, Elm Creek had approximately 37 cars per minute versus the other park had more like 10 to 20, depending on the park. So this suggests that traffic noise effect also depends on the rate of traffic flow, which actually ties into a previous study done by Foreman that found a greater effect of traffic noise with a higher traffic flow. So Foreman looked at various parks where traffic flow differed. So the ones they found a difference in were close to like 25 to 30,000 cars per day. So if we look at our data of Elm Creek, which was about 37 cars per day, cars per minute, and extrapolate that, including like rush hour and decrease, that would be approximately 20 to 25,000, which is nearing that number that could have been associated with an effect of traffic noise. Our other parks, which I mentioned were more like 10 to 20, fell in the range of Foreman's study that did not find an effect on the traffic noise with that traffic flow. Also, Summers, Pollock, and Long found those same edge effects where we decreased abundance as we decreased decibel level as we moved into the habitat and increased abundance as we moved into the habitat. Some potential influences on our study were some counting challenges. So for example, as I'm tromping through the woods, there's lots of brush and buckthorn and prickly ash. Also, there was some flushing of the birds. So as I walked along, I may have scared some birds, and then if they moved to a point ahead of me, I may have counted them again. But missing the birds and double counting the birds, I did my best to keep track of all the birds there. Also, there were some potential weather fluctuations where it was cool and drizzly some days, and also got up to 80 degrees by 8 o'clock in the morning. Also, towards the end of the summer, we saw that wildfire smoke coming down, kind of settling over the Twin Cities area. So this could have affected the bird populations, but also I went to the parks multiple times across the whole summer to help counter this effects. Some potential studies to consider for the future would be looking at, do species individually respond differently to this change in traffic noise? There have been lots of studies done that show that different species respond differently to traffic noise in general, but it would be interesting to see if they respond differently to that change in traffic noise during rush hour. So lastly, I'd like to thank Bethel University and the Seawald and Jones family for funding this project. I'd like to thank Three Rivers Park District for letting me use their parks. Thank you to Jeffrey Ford for advising me on this project and helping me edit it. Thanks to my mom, Jennifer Swanson. I'm pretty sure she's on Zoom. Thanks for coming with me and tromping through the woods and taking some pictures like this turkey here. Also, thanks to my friends Hallie Jones and Ellie Tornell for helping me edit my paper and this presentation. Are there any questions?
So, is there any possibility that traffic noise got in the way at all for you in terms of detecting, being able to detect birds? And we have fewer detections closer to the road. Is there any chance that traffic noise was a distraction for you in being able to detect birds?
Yeah, that's a good question. So the question was, is the traffic noise would the traffic noise have affected my ability to observe birds? And I suppose there could have been a potential for that. The traffic noise, like when I was sitting there, it didn't affect my hearing, I wouldn't think, as much because I could still hear the birds most of the time or see them. For example, Elm Creek was probably the loudest one, but I still didn't have difficulty identifying the birds. I was identifying birds within 50-meter radius of myself, so they were at least like close to me. Does that make sense? You have a follow-up question on that?
No, I have a
Okay. Okay, Dr. Dykstra.
Well, my question is, do you think that the traffic noise is a causation of the abundance that you're seeing, or is it just a correlation, and could there be other factors involved in being close to the road
that would decrease abundance opposed to.
Yeah, so that's why I looked at the edge effects and then also the traffic, like weekend versus weekday. So overall, the edge effects showed that as we looked into the habitat, there was an increase in abundance. But then looking at that specific like traffic noise difference between weekend and weekdays suggests that there might be a cause from traffic noise. However, there are lots of variables because it's nature. So it's hard to know sometimes, but that's where it kind of ties into that traffic flow, where like there are more cars at Elm Creek, so maybe that's why we only saw significance between rush hour and non-rush hour with those trade sects. Dr. Pearson?
So, yeah, I was gonna ask a similar question. So the visual of traffic, for example, would that have had any effect? And maybe noise didn't matter as much. Personally, I think the noise would probably matter more, but I'm just trying to figure out how you can tease out its noise and not just an edge effect that has nothing to do with noise, or visual of traffic that has not necessarily anything to do with noise.
Yeah, so actually there have been studies looking at the visual aspects of traffic, such as like sun reflecting out the windows or like headlights, and they've shown that that has an effect as well. So yeah, there are lots of factors. One way you could possibly control for that is, I don't know, you probably can't have just all the cars turn off their headlights. That might not be good. But if you set up a barrier of some sort that can only block the light and not block the sound, that would be one way to control for that.
Thank you so much. I always feel like there could be so many more questions on and so much more discussion on all of these. Is Jillian here?
Yep, we actually switched, we had to change the schedule a little bit, so she presented already. So that's it.
Oh, so
We're done.
okay.
Yeah.
Well, thank you so much for everybody for attending. I think there's still snacks outside, and excellent job, all of the presenters. Thanks for joining.
No, it was good. And you made it fun.
Dr. Wise says, great work, everyone.